In the fields of molecular biology and genetics, a genome is all genetic material of an organism. It consists of DNA. The genome includes both the genes and the non-coding DNA, as well as mitochondrial DNA and chloroplast DNA. The study of the genome is called genomics. The term genome was created in 1920 by Hans Winkler, professor of botany at the University of Hamburg, Germany. The Oxford Dictionary suggests the name as a blend of the words gene and chromosome. However, see omics for a more thorough discussion. A few related words already existed, such as biome and rhizome, forming a vocabulary into which genome fits systematically. A genome sequence is the complete list of the nucleotides. The next year, Fred Sanger completed the first DNA genome sequence, phage 5x174, of 5,386 base pairs. The first complete genome sequences among all three domains of life were released within a short period during the mid-1990s. The first bacterial genome to be sequenced was that of Haemophilus influenza, completed by a team at the Institute for Genomic Research in 1995. A few months later, the first eukaryotic genome was completed, with sequences of the 16 chromosomes of budding yeast Saccharomyces cerevisiae published as the result of a European-led effort begun in the mid-1980s. The first genome sequence for an archaeon, Methinococcus jannasci, was completed in 1996, again by the Institute for Genomic Research. The development of new technologies has made genome sequencing dramatically cheaper and easier, and the number of complete genome sequences is growing rapidly. The U.S. National Institutes of Health maintains one of several comprehensive databases of genomic information. Among the thousands of completed genome sequencing projects include those for rice, a mouse, the plant Arabidopsis thaliana, the puffer fish, and the bacteria E. coli. In December 2013, scientists first sequenced the entire genome of a Neanderthal, an extinct species of humans. The genome was extracted from the toe bone of a 130,000-year-old Neanderthal found in a Siberian cave. New sequencing technologies, such as massive parallel sequencing, have also opened up the prospect of personal genome sequencing as a diagnostic tool. As pioneered by Mantia Predictive Medicine, a major step toward that goal was the completion in 2007 of the full genome of James D. Watson, one of the co-discoverers of the structure of DNA. Whereas a genome sequence lists the order of every DNA base in a genome, a genome map identifies the landmarks. A genome map is less detailed than a genome sequence and aids in navigating around the genome. The Human Genome Project was organized to map and to sequence the human genome. A fundamental step in the project was the release of a detailed genomic map by Jean Weissenbach and his team at the Genoscope in Paris. Reference genome sequences and maps continue to be updated, removing errors and clarifying regions of high allelic complexity. The decreasing cost of genomic mapping has permitted genealogical sites to offer it as a service, to the extent that one may submit one's genome to crowdsourced scientific endeavors such as DNA Land at the New York Genome Center, an example both of the economies of scale and of citizen science. Viral genomes can be composed of either RNA or DNA. The genomes of RNA viruses can be either single-stranded RNA or double-stranded RNA, and may contain one or more separate RNA molecules. DNA viruses can have either single-stranded or double-stranded genomes. Most DNA virus genomes are composed of a single, linear molecule of DNA, but some are made up of a circular DNA molecule. There are also viral RNA called single-stranded RNA. Serves as template for mRNA synthesis, and single-stranded RNA serves as template for DNA synthesis. Viral envelope is our outer layer of membrane that viral genomes use to enter the host cell. Some of the classes of viral DNA and RNA consist of a viral envelope while some do not. Prokaryotes and eukaryotes have DNA genomes. Archaea and most bacteria have a single circular chromosome. However, some bacterial species have linear or multiple chromosomes. If the DNA is replicated faster than the bacterial cells divide, Multiple copies of the chromosome can be present in a single cell. And if the cells divide faster than the DNA can be replicated, multiple replication of the chromosome is initiated before the division occurs, allowing daughter cells to inherit complete genomes and already partially replicated chromosomes. Most prokaryotes have very little repetitive DNA in their genomes. However, some symbiotic bacteria have reduced genomes and a high fraction of pseudogenes. Only approximately 40% of their DNA encodes proteins. Some bacteria have auxiliary genetic material, also part of their genome, which is carried in plasmids. For this, the word genome should not be used as a synonym of chromosome. Eukaryotic genomes are composed of one or more linear DNA chromosomes. The number O